Foraging Habits by Karson Rosello. Foraging bees and Apis mellifera play a crucial role in the hive. The honeybee's main foraging roles are to collect nectar, pollen, water, and to orient, navigate, communicate, and hive defense. Needless to say, foraging bees have many different roles and are essential for the hive to stay alive. Through their different roles, foraging bees ensure the availability of food resources and contribute to the reproductive success of flowering plants through pollination and maintaining the health and functionality of the hive. However, one of their most important roles is to locate and harvest pollen and nectar. By collecting nectar, the foraging bees search for flowers and produce nectar and store it in their honey stomach known as the crop. When the honeybee gathers pollen, they use their specialized hairs on their bodies to collect pollen and pack them into the pollen brackets on their hind legs. Through learning all of these fascinating things about honeybees, it made me think, how do the foraging habits of Apis mellifera differ based on the different genotypes? Foraging behavior is one of the distinctive behaviors of honeybees, Apis mellifera. This behavior is the link between the honeybee colony and the ambient environment. Therefore, various in-colony and out-colony factors have an impact on this behavior, and many studies have been employed to investigate these factors. Foraging behavior is not advantageous only for the colony and for plant pollination, but also has other benefits. In contrast, some disadvantages have also been discovered to be linked with foraging activities. Practically speaking, the control over this behavior is very important to maximize colony products as well as to increase other agricultural benefits. This paper that I am presenting presents a review on foraging activity, including the regulation of foraging tasks, factors impacting this behavior, foraging preferences, variations between subspecies, and monitoring methods, as well as the possible methods for controlling this behavior. And concluded by this review, it shows that more work needs to be performed in order to elucidate certain aspects of foraging behavior. So I investigated the study that looked at the effects of foraging roles and genotype on light and responsiveness in honeybees. So some background on the study. In honeybees, sensory system can be measured by touching sugar water to the antennae, eliciting the extension of the proboscis. The proboscis extension response, the PER, is closely associated with the complex behavioral traits involving foraging and learning. They were testing whether the sensory motor differences between pollen and nectar specialists are restricted to the gustatory modality or whether pollen foragers are in general more sensitive to sensory stimuli associated with foraging. By testing the responsiveness of varying intensities of light and testing responsiveness of varying concentrations of sucrose in wild-type pollen and non-pollen foragers and bees that are artificially selected for differences in pollen hoarding behaviors. Workers of high pollen hoarding strains are more likely to specialize in collecting pollen. In wild-type bees, pollen foragers are more responsive to sucrose in light than non-pollen foragers. In selected strains, high pollen hoarding pre-foragers were more responsive to sucrose and light than low pollen hoarding pre-foragers. So what they're testing is the sensory responsiveness relating to foraging bees associated with collecting pollen and non-pollen resources, so nectar and water. So they hypothesized a couple things. So they hypothesized that pollen foragers are more responsive to light than non-pollen foragers. 2, high-strain bees are more responsive to light than low-strain bees, which is independent of foraging experience. And then 3, that sensitivity to light is correlated with sensitivity to sucrose in pollen foragers in young pre-foraging bees, supporting the suggestion that sensory modality associated with hoarding and foraging are modulated together. So the results of that were that it found that there's a correlation of the gustatory response scores, the GRS, and the light response scores, LRS, for wild-type bees. The GRS was high for pollen foragers. It was higher for pollen foragers than non-pollen foragers, but light responsive scores did not differ significantly. However, pollen foragers were on average more responsive at low levels of light. And then the figure A is a reference to the median walking response times, so the times that it took for them to respond to the bees in pollen and non-pollen bees. The next experiment that I looked into was the genotypic variation in foraging responses to environmental stimuli. So some background on the experiment is that they experiment a model predicting that colony-level genotypic diversity contributes to colony-level foraging flexibility in honeybees. They established a colony into which they placed individual marked workers from 3 genetically distinct groups. The colony was then placed in an enclosure that contained feeding stations of pollen and sugar syrups. Foraging resources, stores of pollen, and quantity of brood within the colony varied temporally, and then individual foragers switched between resource types in response to changes in the relative resource quality and colony need. This demonstrated flexibility in resource choice at the individual level. However, genetic groups within the colonies varied in their tendency to collect pollen versus nectar and in liability of response to changes of foraging stimuli. So the data suggested that within colony genotypic diversity contributes to resilient foraging responses of environmental variation. So what they were looking for is if there is a significance between the genotypic diversity produced through polyandry. So their method was they assigned workers into 3 genotypic groups, A, B, or C, and depending on their queen. And then the queen and the drone parents for the workers in group A were unrelated and came from 2 unrelated commercial colonies chosen because they contained large quantities of stored pollen. Then the queen and drone parents of group B workers were taken from 2 unrelated commercial colonies with low pollen stores. And then the third queen, Queen C, was naturally mated, and this group was expected to provide a broader distribution of worker genotypes. So the results from this were as predicted by the stimuli threshold models. The genotypic diversity of foraging populations changed in response to variation of foraging stimuli. Genotypic groups with the high threshold for collecting a particular resource increased in representation as the stimulus for the resource increased. We also found genetically based variation in the magnitude and type of individual foraging response to change stimulus in the environment. And they found that within the constraints imposed by genotypically based threshold, individual foragers were capable of flexibility with resource collection. Workers of all 3 genetic groups showed some level of response to changes in internal and external stimulus conditions, resulting in colony-level shifts in resource collection. Furthermore, flexibility in individual resources choices contribute significantly to colony regulation of resource intake, suggesting that there is an important component of colony foraging behavior. So in figure 2 that's shown right here, it shows the proportion of foragers within the 3 genetic groups are collecting pollen under conditions of high and low pollen storage levels within the colony. And then B, so the second graph, shows 25% pollen versus high pollen, pure pollen, resource quality. And then the pollen foraging category include workers that collect pollen, either pollen only or both pollen and nectar. And so the significance of this is the results collected suggested that a functional role of genetic diversity produced by polyandry in colony task regulation by providing behavioral flexibility at the colony level under dynamic environmental conditions. Then I looked into a study that took into account the genotype and rearing environment and how that can affect the honeybee perception and foraging behaviors. So some background, they examined the effects of genotype, the rearing environment, and their interactions on natural foraging behaviors of free-flying bees. The PER of the proboscis extension response, which shows how honeybees perceive and learn about floral odors, which signals the nectar and pollen resources of a colony need for survival. So the experiment, they exposed honeybees from high and low pollen hoarding strains to different pre-emergent rearing environments. High and low larvae were reared in both high and low colony strains environments from hatching to the pupal stage. And then 24 hours prior to adult emergence, the bees were removed from their respective rearing environments and emerged in an incubator. The adult bees were individually marked with the number tags and placed into a common unrelated colony. We tested 4-day-old bees for the response threshold to sucrose using the proboscis extension response assay. The assay began 1 hour after the last bee of each trial was prepared. The assay consisted of sucrose concentrations of which were 0.1, 0.3, 1.0, 3.0, 10, and 30% weight by volume. So the results were the PER score genotype significantly affected the sucrose PER scores, but not the water PER scores. The pre-imagal rearing environment also affected the PER scores. There was a significance genotype by rearing environment interactions for PER scores to sucrose, but not for water. The high-strain bees reared in a high-strain environment had significantly higher PER scores than high-strain bees reared in a low-strain environment. There was no significant difference between low-strain bees reared in different environments. So the significance, this article highlights the significance of how honeybee genotypes in early life environments shape their foraging behaviors. It also highlights the genetic basis of foraging responses to sucrose, which can suggest for potential selective breeding. Then I looked into the effect of genotype on response thresholds to sucrose and foraging behaviors. So some backgrounds on this experiment is they took a look at one of the experiments performed that measured the differences between pollen and non-pollen foragers differing in their response to different sucrose solutions. Foragers were collected directly into small vials taken into a laboratory, segregated from those that had pollen foragers, and those without pollen, non-pollen foragers. They were placed in a freezer, maintained at -15°C, and observed until the first signs of immobility. They then mounted into small brass tubes and assayed for the PER by touching their antennae with a droplet of sucrose solution of water. These techniques were used to describe by Bitterman. Testing began 45 to 60 minutes after the last bee of each group was mounted. Bees were assayed using the concentration series of 0.1, 0.3, 1, 3, 10, and 30% sucrose by weight, corresponding to the logarithmic scale of -1, -0.5, 0, 0.5, 1, and 1.5. All the bees were lined up and tested one time in sequential order, each at the concentrations. All were tested at 0% first, and so on. Bees were always tested with the ascending order of sucrose concentration to reduce potential sensitization that can occur with higher concentrations of sucrose. A bee was then observed to respond by fully extending its proboscis when a drop of sucrose was touched in response to each antennae. So the results before the feedings, pollen foragers were more likely to respond to all concentrations of sucrose than non-pollen foragers. Pollen and non-pollen foragers showed increasing responses to increased concentrations of sucrose, independent of repeated stimulation, as demonstrated by the significant positive slopes of the transformed response curves. Then after feeding resulted in a strong shift downward in total responsiveness to both water and sucrose. Both pollen and non-pollen foragers became significantly less responsive to water after feeding and were no longer different from each other. Non-pollen foragers had a slight but significant increase in their response to water with repeated stimulation following feeding. Pollen and non-pollen foragers were still differed in their responses to sucrose, and response differences between pollen and nectar foragers increased with increasing concentrations of sugar and resulted in differences in the slopes of their respective curves. So the significance is this shows that there are correlation between foraging bees and their sensitivities to different levels of sucrose. In comparison to non-foraging bees, they also outperformed and showed that there was significant difference in the PER, which can indicate difference in genotypes. In conclusion, the foraging habits of Apis mellifera differ based on differing genotypes. The research presented suggested that genetic factors significantly influence foraging behaviors within honeybee colonies. This will then affect preferences, efficiencies, and responses to honeybees' environments and stimuli. So understanding these genotype-specific differences between foraging bees and other bees is crucial for effective colony management and conservation efforts. This proves that specific genes hold promise for different ways in the future that we could potentially breed honeybees as colony loss becomes a real-world issue. The different genome of foraging bees can help us create a future bee colony/PCs that can most effectively find and respond to levels of nectar and pollen on leaves. This will make it easier for honeybees to maintain their hives and help our ecosystem evolve in the future to mitigate future issues of colony loss and lack of space to most efficiently have our honeybees forage the earth. These are my references. And thank you.